Other general things, of course, remember that the exam is on Friday. I will get back the T-cell development problem set to you as soon as I can. I've given you back all of the other graded material. One thing that is a little bit sort of weird in the way that I have the schedule currently set up is for Monday of next week, I have us talking about paper. And that paper is pretty closely related to the stuff we're working on this week. So that paper is posted for next Monday. It's actually related to the Nobel Prize this year. It's one of the papers that was cited for the Nobel Prize. And so that paper and the questions related to it are all posted. They are very closely linked to stuff we're talking about on Wednesday. So at least putting some thought into it may help you remember some of that information, but I am not expecting that you will have looked at it before the exam. But that's there in case you want to look at it. And there will be another assignment due next week that I will hopefully be posting soon, but I had a funny feeling that you're probably not going to be working on it before Friday, what with the exam coming on Friday. So today I'm going to be talking in more detail about T-cell activation and effector function. As I told you last time, when we think about the activation of T cells, we often talk about three key signals that we sometimes refer to as signal 1, signal 2, and signal 3. And last time we went into detail about signal transduction downstream of the T cell receptor, which is signal 1. And so we saw a number of events happening downstream of T-cell receptor signaling, which led to the activation of 3 transcription factors, NF-kappa B, NFAT, and AP1. And the goal of those 3 transcription factors being activated was to allow our activated cell to start making a new protein, and that new protein was IL-2. And so we need to talk a little bit about IL-2 in this whole process. IL-2, as you can see from its name, is a cytokine. It's also shown here is that IL-2. Part of our T cell activation process that is referred to as signal 3, signal 1 and signal 2 are always called signal 1 and signal 2. Then the idea is that there's also cytokines, and some people call that signal 3, but it's not always called signal 3. In any case, IL-2 is part of that cytokines. So that's why sort of where we're talking about it now. This image shows that IL-2 being produced by our cell. Whoa. Yeah, that was spam, and that was terrifying. I thought I had disabled that, but apparently I didn't. Okay. You can see the IL-2 being produced by the T cell and then acting on an IL-2 receptor on that same T cell, which is known as autocrine signaling. And the IL-2 that we see here is often signaling in autocrine. We do sometimes see a few T cells sort of getting activated right next to each other. Maybe the same dendritic cell might actually be activating more than one T cell. And so we can also see paracrine IL-2 from a nearby T cell that's getting activated, giving that IL-2 to that nearby cell. And as I told you last time, IL-2 was previously known as T cell growth factor. And so its key role is to support the growth and survival of T cells. So when our T cell receives IL-2, whether that IL-2 is coming in autocrine, as you can see here, whether it's coming in paracrine from a neighboring T cell, that pushes the cell to go into cell cycle. So we can see several divisions. That also pushes the cell to differentiate, gain some new functions, which are kind of indicated here by the cells now having letters in them, where they didn't have letters in them before. And you can also see this happening here, where our T cell is going to get activated by IL-2 so that it's going to proliferate. It's also going to differentiate and gain some kind of functions. And so as you will see here today, and more so in a bit, the naive T cell doesn't actually really have the ability to do anything about a pathogen. A naive T cell, when it comes out of the thymus, doesn't have the ability to help by making cytokines, doesn't have the ability to kill. It just is still kind of a naive T cell. It only gains that effector function. It only gains its ability to do something about pathogens after it has gone through this activation process and differentiated. And so some of that's happening with IL-2. IL-2 is one member of a family of cytokines. So cytokines can be divided up into different families. And the whole family that IL-2 is part of is pretty famous. So IL-2 is part of a family of cytokines called the common gamma chain family of cytokines. You can see the gamma C in the title here for gamma chain. And the reason why this family of cytokines is classified together is that all of their receptors are multiple proteins, and they all share one protein. So they all share this chain called gamma. And for their receptor to be able to actually, get a signal from that cytokine, they need gamma plus some other chains. So they all have gamma in common. That's why it's called the common gamma chain. You can see the gamma chain here. For the IL-2 receptor, you can see that there's IL-2 receptor beta as well as IL-2 receptor alpha. So it's actually a three-parter. IL-15, you can also see is a three-parter. IL-7 is a gamma chain plus one other chain. IL-9, gamma chain plus one other chain. IL-4, gamma chain plus one other chain. There's also one other member of this family that's not shown here, which is IL-21, which is the gamma chain and one other member. One of the reasons why this is, so here we're seeing IL-2. You guys have already heard a little bit about one of these other cytokines earlier this semester. So which of these other cytokines on this list have you heard about this semester? Yeah, Abigail. IL-7, yes, you guys have heard about IL-7 before. Where did you hear about IL-7 before? Yeah, Abigail. Yeah, so it was in the bone marrow. It was the pat on the head that the B cells got before they made a receptor. It was like for the really early B cells. I didn't say it as specifically, but it actually does the same thing for really early T cells. So the thymus also makes a lot of IL-7 to help the early T cells develop. So we've got this super important cytokine in T cell biology, IL-2. We've got this cytokine that's super important in developing B cells and T cells. And some of these other cytokines actually are important in a lot of other development of other cells. So IL-4 is involved in B cells. IL-15 is involved in NK cells. IL-9 is involved with mast cells. IL-21 is involved with a number of different cells. See Barker et al. So the reason why I point all of this out is that if you were to have a mutation in the gamma chain, or in the gene encoding the gamma chain, and you could not make that gamma chain protein, you would actually lose the ability to respond to all of these cytokines. So if you lose gamma chain, this light blue chain, you lose IL-2 response, and IL-15 response, and IL-7, and IL-9, and IL-4, and IL-21. When that happens, that means you can't have T cell activation. It also means you can't have T cell development or B cell development or NK cells or mast cells. And so you actually have pretty broad immunodeficiency. You are actually basically have no adaptive immune system if you are missing this one protein, because it is shared as a receptor chain by so many different cytokines. And this is actually one of the most commonly seen immunodeficiencies. So I think I showed you before the little boy in a bubble. That boy has a disease called SCID, which is severe combined immunodeficiency. And he is lacking the common gamma chain. So he cannot make a response to any of these cytokines, so he loses all of these different immunological processes. The gamma chain is encoded on the X chromosome, making this an X-linked disease, something that we frequently see in young boys. And so this family of cytokines is sort of like the particularly important family of cytokines. It all involves these different types of receptors. You can also notice, if you look at the IL-2 receptor, as I've already pointed out, the IL-2 receptor has 3 different chains. It has the gamma chain that is shared amongst all of these cytokines. It has another chain called the IL-2 receptor beta chain, and one called the IL-2 receptor alpha chain. And so the best IL-2 receptor has all 3 of those. You can see that if we have gamma and just the IL-2 receptor beta chain, that can start to become a receptor for IL-15 as well if it's paired with a different alpha chain. And so you could imagine a version of this receptor with just gamma. You could envision a version that has gamma and beta, and you can envision a version that has, you can envision alpha, beta, gamma, you can envision beta, gamma, you can envision alpha. Yeah, you could envision other combos as well, but these are the ones that I want you to look at. And all 3 of the versions that you see here, whether it's alpha by itself, beta and gamma by itself, or all 3, alpha, beta, and gamma, can bind IL-2 and get some kind of signal through IL-2. You can see that alpha by itself is the weakest. If you have beta and gamma, you kind of have this intermediate. If you have all 3, you bind IL-2 super well. You like suck up all the IL-2 in the room. And you are going to get the best IL-2 signal. Because this will be interesting later, especially in the thinking about that paper for next week, especially in answering one of the questions for that paper for next week that I just wrote right before I came here, I will just point out that these proteins do have other names. So the gamma chain is also known as CD132. The beta chain is also known as CD122. And the alpha chain is also known as CD25. And so what you can notice here is if you have a cell that sort of adds CD25 to a beta and a gamma, then it becomes really good at binding IL-2. Way back when we talked about T cell development and DN1 through DN4 stages, CD25 was one of the things that was used to mark some of the types of developing T cells. And there are also some other places where people use CD25 to mark cells. And so that's part of, I really don't care that you know about 132 or 122, but CD25 being the alpha chain is sort of the more important one. And the idea, the thing that's sort of relevant with this is that when our T cell is naive, it only has beta and gamma on its surface. But when that T cell becomes activated, when that T cell actually starts making IL-2, it also starts making IL-2 receptor alpha, otherwise known as CD25, and being really good at sucking up IL-2. So, you can imagine when it first makes an IL-2 molecule, it can sort of be okay at binding that IL-2 molecule. But then as time goes on, it's going to be like, yeah, give me that IL-2. And we're going to start to have a much better receptor, and we're going to kind of start sucking up that IL-2. And so you can kind of see this, I think, here as well, where I guess not, but I have a version somewhere in all of the slides I own where you can see that the cell starts out sort of earlier, making the medium affinity IL-2 receptor. Oh, I see it now. You can see that at the beginning, the cell is kind of making the medium affinity IL-2 receptor. Eventually, the cell is going to start making IL-2. It's also going to start making the high affinity receptor, and it's going to start feeding itself IL-2, now using this super good receptor, and now it will divide. And this is, I flipped the order of a couple slides, so that's where this slide is if you want to look at it. Yeah, Jazz.
So we discussed where we'd see gamma, beta. We discussed where we'd see all three of them. Where would you see only alpha?
I cannot think of a obvious situation on the top of my head. Okay. And so I would not get super stressed about that. Okay. There is one place I'm debating in my head, and I don't want to go down that rabbit hole right now. So I will just say that right now I would think about either beta gamma versus all three. Okay. And so what I want you to be thinking about here is the idea that when a T cell is not active, it has only beta and gamma. Once we start this activation process, it has alpha. And so you could imagine alpha could be a way to tell you, was my T cell activated or not. You could look and see, does my T cell have alpha on it? That might be one way that you could say, this T cell is activated compared to another T cell. Does that make sense? Okay.
Thank you.
Yeah. Just like we talked about signal transduction downstream of the T cell receptor, I also want to tell you a little bit about signal transduction downstream of the IL-2 receptor. I'm not actually going to tell you about like super specific answers to the IL-2 receptor. I'm actually going to tell you general cytokine signaling. So generally how signaling downstream of a cytokine receptor works. IL-2 could be one specific example, but there could be many others. If you remember last time we talked about the RAS-MAP kinase pathway, and I told you if you could remember the name of it was the RAS-MAP kinase pathway, you were pretty good in that you pretty much knew what was going on just from the name RAS-MAP kinase. the signaling pathway that we see with cytokines is similar in that if you can remember the name of this pathway, you're doing pretty good. So to start with, whether we're talking about the IL-2 receptor or whether we're talking about any other cytokine receptor, we've got some different chains that are diffusing away from each other in the membrane. And when the cytokine binds, they're brought together through induced proximity. So of course we induce the proximity of the cytoplasmic tails as well. And our cytokine receptor always is found, or all cytokine receptors are always found with a partner protein attached at to their cytoplasmic tail. This partner protein is called JAK, J-A-K. Officially, there are 4 different JAKs: JAK1, JAK2, JAK3, and TYK2. But we're just going to say JAK right now. Which JAK it is dependent on which cytokine we're talking about. We're just going to say it's a JAK. And so you can see we've got the JAKs associated with the cytokine receptor. You can see them coming into induced proximity when the cytokine binds. And the JAKs, the K in JAK stands for kinase. So the kinase will allow phosphorylation, and basically we've brought these 2 kinases next to each other so they phosphorylate each other. So you can see the phosphorylation happening of these kinases now that they are brought together. Just like we saw with signaling last time, once we have stuff getting phosphorylated, that results in a new binding site in the cell. And so now we can have our next partner bind, and our next partner is called STAT. And so you can see STAT binding to those phosphorylated JAKs. The STATs will also get phosphorylated, so you can see the STATs getting phosphorylated. There are 7 STATs. 1, 2, 3, 4, 5A, 5B, and 6. Again, when we're talking about which cytokine is going on here, it matters which STAT and which JAK, but here we're just going to call it JAK and STAT. As you can see, the STATs get phosphorylated. When they're phosphorylated, they bind together. You can see that here. And when they bind together, they are able to go to the nucleus and be a transcription factor. That is the whole pathway. So this pathway is sometimes called the JAK-STAT pathway. If you can remember, this pathway is called the JAK-STAT pathway, you know the whole pathway. STAT, as you can see up at the top, stands for signal transducer and activator of transcription, because it both transduces a signal and is a transcription factor. So that's kind of a useful name. JAK's name is kind of funny. So if you look at JAK, and if you think about what we were talking about with receptors, when we talked about other receptors earlier, JAK clearly has the ability to phosphorylate things. It's got a part that can do phosphorylating. That's where the K's comes from. It's kinase. And you can also see it has the important role of getting phosphorylated, right? So it has a part that can get phosphorylated and has a part that does phosphorylating. So it has like 2 different sides to it. So the story goes that JAK was named after the Roman god who had 2 faces, who is Janus. So it is the Janus kinase, because it has 2 faces, one that gets phosphorylated and one that does phosphorylating. You can also see that JAKs would be important in diseases like SCID. In fact, there are some patients who have JAK mutations that get SCID. And so I am told by super old school immunologists that the name Janus kinase, sounding all nice for the two faces of JAK, is a lie. I'm told that that was not originally what it was. I was told that they made that up later to make it sound nice when they knew more about it. Originally, they tell me that they thought they had found something super interesting in immune regulation, and then they realized they had found just another kinase. And so they say that JAK originally stood for just another kinase, and then they retroactively made it Janus kinase to sound fancy. The other thing I will say regarding this is one of the big new drugs that is used in a lot of patients with autoimmunity right now are JAK inhibitors. So JAK inhibitors are like the new hot thing. They actually have a lot of side effects, but they are pretty useful in patients for whom other drugs won't work. And so obviously to go into more detail with that, I'd have to tell you about specific JAKs, which I am not going to do here because those inhibitors that different patients use inhibit specific JAKs. But this is the situation about JAK-STAT. So the big thing that we need to talk about today is the other piece of activation of T cells. And we saw a lot of information about signal 1. I just told you some stuff about signal 3. You'll see some more stuff about signal 3 on Wednesday. But I haven't told you about signal 2. And signal 2 is super, 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 I don't have enough capital letters, important. And so we really need to spend some time thinking about signal 2. Now, you could say that I have arranged this section of the course the way I had it on the syllabus, and I'm telling you about the things in order that I was planning on telling you about things, and you'd be right. But you could also say and note that the last time Emily was here, she left us extremely worried about T cells leaving the thymus and how we have so many self-reactive T cells leaving the thymus. How every T cell that leaves the thymus is a little self-reactive. And she's like, but I want to know about peripheral tolerance. What's going on with peripheral tolerance? She really wanted to know about peripheral tolerance. Signal 2 is a huge part of the answer to Emily's question. Signal 2 is one of the big answers of central tolerance, or peripheral tolerance, excuse me. It's not the only peripheral tolerance answer. There are actually multiple, and we have a whole peripheral tolerance lecture that we'll see later on. But signal 2 is actually a big piece of it. So first we can just kind of see the basics of signal 2 on this slide. So what you can notice for signal 2 is that it's shown in this figure on the left as this separate protein-protein interaction between the T cell and the antigen-presenting cell. And if we look at this image from your textbook, it correctly even gives us names for those proteins involved in the protein-protein interaction. It calls the cell on the antigen-presenting cell, in this case the dendritic cell, B7, and it calls the protein on the T cell CD28. And so we tend to see our T cell getting signal 2 through this protein called CD28 if that dendritic cell is making this protein B7. So we're going to be thinking about a dendritic cell is going to be making B7. That's going to give a signal to CD28. And in a few slides, we're also going to think about what happens if there is no B7. What happens if we get a T cell receptor signal, but we don't get signal 2? So CD28 is left hanging because there's no B7. And this is sort of the key piece to all of this. And so one thing that I will say is that to activate a T cell, in particular to activate a naive T cell. The first time the T cell is getting activated in the lymph node, that very first time the T cell is getting activated, signal 2 is required. Again, I don't have enough capital letters. I don't know how to say capital letters. But signal 2 is required for a naive T cell to be activated. That T cell must get a signal through CD28. That signal through CD28 is coming from this other protein called B7. And if you remember back a ways, I told you that dendritic cells were really, really, really good at initially activating T cells. I told you that dendritic cells were the stars of initial T cell activation. That's because dendritic cells are so good at making B7. And so what we need to think about, what we need to think about is why exactly. In particular, why exactly is our dendritic cell going to make B7? Because what you can notice here is that if our dendritic cell doesn't make B7, this doesn't work. Our T cell doesn't get activated. So we have to think about, well, what is going to make our dendritic cell, or any antigen presenting cell, but most frequently a dendritic cell, why would it make B7? And the answer is that making B7 is a key part of dendritic cell activation. So when a dendritic cell is activated, one of the biggest things that happens is that dendritic cell starts to make B7. And so you can see before this, our dendritic cell did not have B7. Once our dendritic cell is activated, you see that dendritic cell starts to make B7. I just realized that between today and Wednesday, there are 2 areas of immunology that were, like when I was an undergrad, I was taught that they were like forbidden, wrong, like no one thinks about them, and if you ever talked about them, like they would strike you down. And they have both become like super commonplace, and like everyone talks about them now. They're like totally a thing. And I just realized I think I'm gonna be struck by lightning both today and Wednesday, but that's okay. So what we can think about, what we now know, is what was the key event that helped that dendritic cell be activated? How did the dendritic cell know it was time to be activated? Yeah, Abigail. So there was pathogen binding to a PRR. Remember, we had a pathogen bind to a PRR that made the dendritic cell become activated. And in fact, when I showed you this slide way back at the beginning of the semester, I showed you, oh look, there's a PRR. And as a result, the cell makes cytokines. The cell also makes B7, this co-stimulatory molecule here. And so what you can imagine is that this is the theory that is now totally accepted, but was back in the day a struck by lightning thing. The idea is you can kind of think about this PRR sensing a MAMP as sort of like a danger signal. It's something to say, hey, dendritic cell, there's something foreign around. And so this is allowing the dendritic cell to say, you know what, I was in some kind of dangerous situation. I saw some sort of microbe. Whatever I'm presenting on my MHC might be bad. It's sort of this additional signal that is giving the T cell to say, hey, T cell, there's danger. So it's both, here's an antigen, and that antigen is dangerous. As opposed to if there was no B7, that probably means there was no PRR, no pathogen stimulating a PRR. So this antigen in that case might just be a self-antigen. It might not be a dangerous antigen. And so we have this important way that our antigen presenting cell can say, hey, I was activated by like innate immune receptors. I think there's something going on. I really think you need to be ready for this thing, antigen I'm presenting. So people often will talk about the B7 as almost like as the danger signal that the antigen presenting cell is giving in addition to antigen, to kind of give some context about that antigen. Did you see me not get struck by lightning when I said danger? And so for a T cell to be activated, that T cell must see signal 1 and signal 2. And again, this is the first time the T cell is seeing antigen in the lymph node. This is a naive T cell getting activated its first time. So that's one key thing to think about, is we gotta have B7. Also, if we're being technically correct, there are two B7s. They're B7-1 and B7-2. They are also known as CD80 and CD86. You may see them labeled with these specifics on some slide or something. As far as I'm concerned, you can just call B7. If you look back at the Medzitov paper that you read earlier in the semester, they were measuring B7 production following signaling as one of their measures there. So that is really important. So like I said, one thing that I cannot emphasize enough, first time a T cell is activated, coming out of the thymus, makes it to the lymph node, getting activated the first time seeing antigen, must have signal 2. Must use CD28 on that T cell to bind to B7 on this other cell type. What's going on in a situation where a T cell sees antigen with its T cell receptor but doesn't see B7? So CD28 gets nothing. There's no B7 around. CD28 has nothing to bind it. And so we're getting signal 1, but we get no signal 2. What's the situation where that could happen? Yeah, Jazz.
Binding to self.
So that might be binding to self. In particular, that might be, remember how we've talked about the idea that all cells are presenting self, especially on class 1, all the time, right? So they're going to be presenting on class 1 self all the time, but they're not going to be presenting or making any B7. So if a T cell happens to come across its antigen without seeing B7, it's probably seeing a self cell presenting self antigen, right? It's seeing kind of that background self cells just showing what they got. And this is happening in the periphery. So if a T cell is seeing kind of self antigen on just a normal, happy, healthy cell, it's going to see signal 1, but it's not going to see signal 2, right? What do you want to have happen in that situation? What should be the goal in that situation? You got a cell, it leaves the thymus, it's seeing its antigen on self stuff. Yeah, Emily. Okay, you want it to let it be and not do anything. Yeah, David.
Want it to be deactivated.
Yeah, actually what you really want is you want that cell to be deactivated, because now it is actually in the periphery being a little dangery. So you would like that cell to get turned off. You would like that cell, now that that cell is actually able to get a signal in the periphery without B7, you're like, yeah, how about you just shut it down, right? And so in fact, if a T cell sees signal 1 without signal 2, and you can see an example here, signal 1 without signal 2, that T cell is going to go to anergy. And so we saw anergy with B cells It's this state where the cell is basically turned off and unresponsive. And so if we have some cell that's presenting self-antigen, the T cell recognizes it, we're going to energize that T cell. We're going to be like, no, you need to shut down. And so this is going to be a form of central tolerance. So you can basically imagine every single one of those autoreactive T cells, those self-reactive T cells, as long as they see their self-antigen somewhere in the body, as long as the first thing they see when they leave the T cell is their antigen without any B7, they get shut off and they're shut off for good. The problem happens in the rare situation where they see something else first, which we'll get to. But the normal situation is as they're just circulating around the body, they're going to find their antigen on signal 1. There's not going to be signal 2. Part of this is because many other non-immune cell types aren't very good at making signal 2. And they're going to get turned off, is the way that this process is going to work. Yep, Jazz.
I think we did.
Okay, Paige first.
So B7 is only on the dendritic cell if it's like not healthy?
If it's activated. So only when the dendritic cell is activated. And so the way the dendritic cell gets activated is that it's seen a microbe. Yeah. Yeah, okay. So Jazz, did you have a question first?
Why is this considered central tolerance and not peripheral?
It's peripheral tolerance. I'm sorry if I said that incorrectly. It's peripheral tolerance. Yep, Emily.
Would it be fair to say B7 is kind of like a bridge between innate and adaptive?
In some ways, you can think about B7 as being a bridge between innate and adaptive. And because you can see how important this is. This is basically how it is that you can have all these self-reactive T cells around, because they all need B7 to get turned on. And that is this bridge between innate and adaptive immunity. This is why the 2011 Nobel Prize was given for this innate adaptive immunity bridge. So you can see kind of the key importance here, and it is so crazy important. Yep, Chrissy.
B7 only made by dendritic cells?
It is not only made by dendritic cells, but it is made by dendritic cells by far the best, and at the highest amounts. So sort of as we can kind of see here, the reason why we do all of this is because every T cell that comes out of the thymus has the ability to react to self-antigen a little bit. So every T cell that comes out of the thymus, every T cell that is positively selected, needs sort of this extra check on it, this extra break of B7 in order to be first activated. And you might wonder, well, why do we do positive selection this way at all? Like this I'm still not loving this whole, all these self-reactive T cells thing. Why do we do our selection like this at all? And really the answer is, if you think about it, if you think about what a T cell is seeing, it is seeing a small number of amino acids from a microbe. It is, again, I'm a CD8 girl, so I often think about class 1. So it's seeing 8 to 10 amino acids of our microbe, right? And it's seeing it with relatively low affinity in the thymus. Okay, if you're only seeing 8 amino acids, a little string of 8 amino acids, like how many variations of things are there that you can see? There's only 20 amino acids, so there's like 20 choices at 8 places. It's not like your ability, you can see like literally every possible thing in the world. And oftentimes, if you just change like one of those 8 or something, you might go from low to high affinity. Because basically biochemistry has sort of limited us to a relatively small set of amino acids that are used to make both pathogens and us, if we do selection on self-peptides for things that are low affinity, they are likely going to be relatively similar to things that would be from a pathogen. They might be only a teensy bit different, and that teensy bit different might be enough to make them high affinity. Another way of saying this is that many pathogen peptides are extremely similar to self-peptides and can be cross-reactive. So a T cell that recognizes self is generally also recognizing a similar pathogen peptide. And this shows you some sort of examples of this. So here is a peptide from measles that can be recognized by some T cells. This is a peptide from myelin. And so you can see that the self-peptide and the foreign peptide are actually pretty close. It's only a pretty small biochemical difference. Similarly, here's one for HLA-B27 itself and a Klebsiella protein. And you could go through this on and on and on. And so by and large, many microbe proteins look really similar to many self proteins. So if the T cell sees its self protein, it's going to get turned off. If the T cell sees it, and it's probably going to be relatively low affinity. The T cell sees the microbe protein, it might be higher affinity, and it's also going to get B7. And so this is the other reason why we have to have this sort of additional check. If you think about it, this HIV protein actually has the exact same peptide as IgG itself. And so we really want to make sure that the T cell only binds to HIV and not to any B cell. And so we do that because the T cell, when it sees an HIV-infected cell, is seeing signal 1 and signal 2. While if it's only binding to a B cell and seeing this as a self-antigen, it's seeing signal 1 without signal 2. So oftentimes we get the affinity bump from the sort of cross-reactive amino acids or the slightly different amino acids, but we also have sort of that B7 thing. And so this is also why that B7 is so important. And so if we have a T cell that first sees its antigen without B7, we sort of imagine that that is antigen being seen in a self-presentation context, because there was nothing from a pathogen to turn on B7. And we are going to turn that T cell off, theoretically permanently. Maybe not permanently. If there is like an absolute sledgehammer of an immune response happening, if you are in like a super emergency situation, that T cell can get turned back on, but only in a crazy emergency situation. So imagine it being off. It is on. It is still turn onable in an emergency situation, because you're like, in an emergency, we want to have everybody. Even if we might get autoimmune disease, at least it'll kill the pathogen. But outside of that emergency, that cell is off. This information is super important to know to understand the peripheral tolerance question that Emily was asking about before. It's also super important if you are an immunologist who wants to work with T cells in the lab. If you want to work with T cells in the lab, you actually can't turn them on if you don't give them signal 2. If you just try to give them signal 1 in your dish without giving them signal 2, you are going to make a whole bunch of anergic T cells. And that is not what you want, probably. And so, in fact, you have to actually give the cell something to trigger signal 1 and something to trigger signal 2. Oftentimes, we either use a chemical signal 1 or we use an antibody that actually hits CD3 to give it a signal. So it sort of like fakes the cell out by thinking it's getting a CD3 signal using this antibody. And we can use the same kind of thing with an anti-CD28 antibody to fake the cell out. I will note there are different antibodies against CD3, different antibodies against CD28, different antibodies against all the things. These are what are known as activating antibodies. There are also antibodies that exist that block. So you could have a different CD3 antibody that blocked and made it so that no one else could interact with CD3. And so you never got a CD3 signal. You can also have a CD28 blocking antibody that blocks CD28, and so you could never get a signal 2 through it. This is an activating CD28 antibody. And so in fact, the only way we can get T cells to proliferate is if we give them signal 1 and signal 2, through both some way of giving them a signal 1 and some way of giving them a signal 2 activation. And so what we were just talking about was about the activation of a naive T cell. And so you can see our naive T cell being activated on the left, and our naive T cell needing signal 1 and signal 2. This is in the lymph node. This is turning that T cell on, starting that T cell to divide, telling that T cell to start doing its effector function. The T cell does not yet have an effector function. This is a purple T cell, which in your textbooks thing is CD8. Also, I like CD8, so yay. It's a CD8 T cell. Remember, the CD8 T cells are the ones that are the killers. This T cell doesn't kill the dendritic cell it first sees. It hasn't figured out how to kill yet. It hasn't been activated and differentiated to kill. After this interaction, it's going to become a killer T cell. It now is going to have the ability to kill. So it is no longer a naive T cell. After this, it's going to be an activated T cell. When that T cell is an activated T cell, it's going to go to some other body site. One of the ways that it's going to do that process of going to some other body site was a slide that I really thought was next and definitely is not. But it's going to involve changes in trafficking molecules. So the cell is going to change what its trafficking molecules are so that it can go somewhere else, like to the lung in case of SARS-CoV-2 or to the liver in case of hepatitis B. And now that T cell is going to be able to kill in that location. Importantly, when this cell is activated, when this cell has been turned on, it no longer needs signal 2. It only needed signal 2 the first time in order to get turned on. Once that cell is turned on, it's good and can just keep killing anything it sees with its antigen, with signal 1. So that's part of, this relates to Chrissy's question. It relates to a bunch of things. But so this is part of the idea of when that cell goes elsewhere, it doesn't need signal 2 anymore. It only needs signal 1. The signal 2 requirement is only for the activation of the naive cell in the lymph node. So the other thing that I have to tell you about is that in talking about signal 1 and signal 2, we've been talking about CD28 on a T cell binding to B7 on an antigen presenting cell. And that is sort of the famous signal 2 co-stimulatory situation. But in reality, there are tons of options for co-stimulatory molecules, both on the T cell side and the antigen presenting cell side. B7 CD28 is always needed for naive T cells, and it's real famous. But when the T cell is maybe activated, it might use a different type of co-stimulatory molecule. And so what I want you to see here is that there are tons of co-stimulatory molecules, and this side is showing us the antigen presenting cell in sort of pinkish. This yellowish is showing us the T cell. You can see what I want you to see. All I want you to see is there's lots of choices. So there are lots of other ones other than the ones I'm telling you about. I'm just telling you about the one that is the most famous. There are 2 other co-stimulatory molecules that are famous in certain situations. And so we need to also talk, while we're talking about these co-stimulatory molecules, about the 2 other ones that can be famous in particular situations. One of them is called CTLA4. You can see CTLA-4 listed here. The other one is called PD-1. And so I'm going to tell you a little bit about CTLA-4 and PD-1. In particular, I'm going to say much more about CTLA-4, and then I'll say a little about PD-1. You can see CTLA-4 and PD-1 right up here alongside CD28. So here's CD28 on my T cell. Here's CTLA-4. Here's PD-1. And if you look at this image over here on the left, it's also said in words. If we look at CD28 and we look at PD-1 and CTLA-4, what do we notice is different between them here? Yeah, Roxy. Okay, so you're saying charge. Basically, there's a pluses and minuses. It's not actually charge. It's actually what happens to the T cell if it gets a signal. With CD28, it turns the T cell on. With the other ones, it's turning the T cell off. So it's actually, is it a negative, or is it a stimulatory signal, or is it an inhibitory signal? Are we turning this T cell on, or are we turning the T cell off? And CTLA-4 is definitely our most famous of the inhibitory signals. And so you can see on the left a situation where a T cell is binding to T cell receptor and CD28, getting signal 1 and signal 2, and that is activating the T cell. You can also see, I'm hiding this for a second, a situation where the T cell is getting signal 1 through T cell receptor, and it's getting some binding through CTLA-4. So whatever CTLA-4 binds to is getting ligated, and we're getting a signal here, and that is actually going to inhibit the T cell. That's an off switch for the T cell. And you might wonder what CTLA-4 is binding. It turns out that CTLA-4 also binds B7. And CTLA-4 binds B7 much more strongly than does CD28. So in a situation like the situation you see here, where there's a choice between CD28 and CTLA-4 binding, the B7 is going to bind the CTLA-4 and not the CD28. So basically, if a T cell happens to have both of these, both CD28 and CTLA-4, B7 is going to turn that T cell off instead of it turning it on. It's not going to energize it. It could be turned back on later. This is just an off for now, like a light switch. It could come back on. Yep, Jazz?
So if the binding of B7 to CTLA-4 is stronger than that of its binding to CD28, how does this become temporary? Like how does B7 connect to CD28 yet? Oh, amazing.
So when our T cell is naive, that T cell only has, this is not labeled, but it should be, that T cell only has B7. So when that T cell is naive, it's only got B7. You can see it'll eventually start making things like IL-2 receptor, CD25. So you can see our T cell sort of getting activated here. Once our T cell gets activated, it also starts making CTLA-4. So basically, when we turn our T cell on, the T cell also gets itself an off switch. So the T cell doesn't start with the off switch. It doesn't start with CTLA-4, but it turns on CTLA-4 when it's like, okay, I could be too much. Turn me off if I'm too much. It's basically automatically turning on the brake when you are activating the cell, so that once that turned on cell can get turned back off. Eventually, CTLA-4 may go away, and so then you can activate that cell again because CTLA-4 expression will go away. Does that answer your question? Okay. And so what you can see is that some of these proteins that we are talking about here vary on cells that are at different stages of their life. You can imagine if a T cell started making too much CTLA-4. let's imagine for whatever reason, a T cell just started making CTLA-4. What would happen if a T cell just started making CTLA-4 randomly? Or if you just had too much CTLA-4 in general. Yeah, Paige. It would never be activated. You'd basically have that T cell off all the time. There are certain situations where we can see T cells making too much CTLA-4. And in that case, the immune system isn't working right. We don't have good T cell activity in certain cases. It specifically, this will come up much more later, many tumors will push T cells to make CTLA-4 and be like, stop killing me, T cells. Then the T cells will start making CTLA-4 and the T cells can't kill the tumor anymore. The T cells can no longer be activated to kill the tumor because we've started getting excess CTLA-4 production. So this is all particularly important. There is a similar inhibitory molecule called PD-1. PD-1, and so at the top here you can see, here's my naive T cell. It's interacting with a dendritic cell. It's getting signal through signal 1, and it's getting signal through signal 2. Now once that T cell gets activated, we start getting CTLA-4, so we turn that T cell off. If that T cell is sort of later on in a different point of its life, or if we're in different types of tissues, one of the negative signals might be one called PD-1 on the T cell that is going to bind to something called PDL1. And so you can see PDL1 binding to PD-1 here. It's having the same effect on that T cell. It's just that this might be in a tissue, or I usually think about it as later in the T cell's life. When people started to look at a bunch of different situations with T cells, they found that there are certain situations where T cells do not work very well. One of them might be in a cancer situation. One of them is a situation I think we can all understand pretty well right now. So let's imagine if you are a T cell and you respond to influenza virus. Now, we're going to say, I'm going to tell you influenza virus, your body can get rid of influenza virus in like 2 days. I'm just making that number up, a couple days. So you're a T cell, you respond to influenza virus. You come in, you're a T cell, you respond to influenza virus. So then what are you going to do, T cell, if I'm influenza? You're going to get activated. You're probably going to get rid of me, right? Then what are you going to do after you get rid of me? Inactivate, chill, take a nap, lay on a hammock. Once upon a time, I laid on the front table as a hammock in this situation. Okay? Now let's imagine we are responding to a herpes virus. Herpes viruses are your body cannot ever get rid of them. It's in your body for the rest of your life. This would also be true if it was a tumor, which you're going to have for the rest of your life. This is also true for a lot of things. Those are the two, I told you 2 common ones, but there's actually a bunch of them. All right, you are responding. Imagine that you are responding to this herpes virus that plagues you and will not go away. What are you going to do? Activate. You're going to try to kill that virus. Are you going to succeed? No, you're never going to succeed. So what are you going to do? Again, think about this part of the semester. Stay activated. Keep working real hard, keep working real hard, keep working real hard, keep working real hard, keep working real hard. Then what's going to happen? Yeah.
But then you get tired and exhausted.
Then you get tired and exhausted, right? In those situations, there's actually something called T cell exhaustion, where the T cells are actually acting for so much that they actually just sort of stop, almost like give up. One of the things we notice when T cells sort of stop or give up, or when they're exhausted, is they have a lot of PD-1 on their surface. So this PD-1 negative signal is sort of involved in T cell exhaustion. So you can see, here's some T cell exhaustion. And basically what you can see is this is a T cell that's getting more and more exhausted at the bottom. and it's getting a lot of PD-1 on its surface. So T cells, as they get really exhausted, they never get turned off. They're going to get PD-1 on their surface. Yep, Emily.
So why, like, what's the purpose of CTLA4 and PD-1 if, like, you don't need to be activated?
So, excellent question. I'm just trying to think how, like, where, because it's a great question.
Like, if you see your pathogen, then you get activated, you take care of that, and then you're immediately like, let me turn off, even though, like, cells everywhere around you could also have the same pathogen.
Okay. I'm trying to think how to explain it. No, it's like such a key thing. All right, so no, where is it? Emily is going to be measles virus P3, okay? And Nadia is going to be myelin basic protein, or actually corticotropin. So they're really, really similar, okay? So let's imagine I'm a T cell. I'm a naive T cell that binds to this epitope, okay? Here I am, naive T cell. Naive T cell, naive T cell. I come across Nadia. What's going to happen? I'm a naive T cell. Nadia is a cell expressing corticotropin, which is a self-peptide. I'm going to get energized, right? I'm going to get signal 1, no signal 2. And so I'm going to get energized because that's what I needed, right? Okay? Here I am, naive T cell, naive T cell, naive T cell. I interact with Emily. What am I going to do? I'm going to get activated, right? And I'm going to go somewhere else in the body and kill all the measles, right? What happens if I come across Nadia?
You're activated.
I'm activated. I don't need signal 2 anymore. I'm now going to kill self-antigens. Basically, if T cells are on too long, they start to cause problems. They start to like wreak havoc and basically start attacking self. And so there are a lot of cases actually where if we don't turn off a T cell response at the right time, if we don't contract that T cell response pretty soon afterwards, if we just leave the T cells on forever, there are a bunch of problems that happen. Okay. One of them is how much glucose they're consuming and sort of things like that. But one of them, how much space they're consuming or taking up in the lymph node. But it's also that they can actually start attacking self now. Because you've taken off the brake, they don't need signal 2 anymore. Okay? So it's actually super important that once they're done, you turn them off. Okay. And that's also going to come up again more later. Yeah. No, that's such a good question. That's why I was like, I have to do this right. So related to that, some investigators realized, hey, look, we've got certain situations where T cells are getting too much negative signal. They're getting too much PD-1 signal, or they're getting too much CTLA4 signal. In particular, say T cells that are responding to cancers, but also T cells that are responding to chronic infections like HIV. T cells that are responding for really long periods of time. And so these investigators came up with a hypothesis for how they might be able to take advantage of this situation. So can you imagine a hypothesis for what you might do here? It's actually a little bit tricky after having answered Emily's question because you're going to obviously see an issue with it, but it's okay, we'll get there. Yeah, David.
What if we stop expressing PD-1 on those tired cells?
Okay, so one thing we could do is we could stop expressing PD-1 on those tired cells. We don't really know how to make the cells stop expressing PD-1, but yeah. What if you put in something that could block PD-1? What if you put a little cap on PD-1 so it couldn't get a signal anymore, right? So the idea is what people started to use is they started to use blocking antibodies. They used a blocking antibody against CTLA4, so CTLA4 could never get triggered. We could never turn the T cell off. Or a blocking antibody against PD-1. Again, so we can never get that negative signal on the T cell. The T cell never gets turned off. You keep the T cells on forever. This is checkpoint inhibition. This was the Nobel Prize in like 2020. I can't remember what year it was. But in certain cases, we will actually use an antibody against CTLA-4 so that CTLA-4 can never get triggered. You can never turn the T cell off. And so there are checkpoint inhibition drugs that work by blocking CTLA-4. There are others that work by blocking PD-1, which you can see here. You may have heard of Keytruda or Opdivo, which are key cancer drugs. Yeah, they're blocking PD-1. They're the PD-1 blocks. And so blocking PD-1 and blocking CTLA-4 are actually really key in situations where we have T cells that have stopped working, and we want the T cells to get their act together and start working. So we block PD-1 or CTLA-4. There is a potential complication to these drugs. What complication might you imagine for these drugs? Yeah, Jazz.
Emily's instance,
Which is?
which is when you're overactivated and when you have the T cell exhaustion.
Yeah, in immunology, we are pretty good at Goldilocks. We're pretty good at too much or too little. And so if you have too little T cells, we can make you have more, but you might end up with too much in autoimmunity. So if you're treated with these drugs, you might actually get the T cells to stop your cancer, but you also might get autoimmune disease. You got to think about the trade-offs there of, well, if you were going to die of the cancer and you're going to live for 20 years with the autoimmune disease, which one's better? It becomes a trade-off. We can't actually block just the one specific T cell at this point. We can either kind of block them all or only block some. And so at this point, our T cell is activated, and that T cell is in the periphery. And so you can see the steps of activation that we've been talking about, signal 1 and signal 2. We can see some cytokine production. We can see clonal expansion, where that cell is making a clone army of itself. It's got pat on the head. It's now divided a lot. We saw mitosis. So we've made many copies of that cell. And that cell has now learned how to do its function. So it finally is going to do something against a microbe. It's going to probably do that in a tissue. It's no longer going to the lymph node, so it's going to change its trafficking patterns and go to the tissue. And it has different signaling requirements. It doesn't require signal 2 anymore in the tissue, so now it's going to do something and get rid of the pathogen. And on Wednesday, we will get rid of the pathogen and see the effector functions of T cells. So I will see you guys on Wednesday.